Unlocking human DNA, the role of ancient viruses. Imagine discovering that ancient viruses in our DNA could unlock the secrets of our humanity. Sounds intriguing, right? Well, a groundbreaking international study suggests just that. Researchers from McGill University and Kyoto University have found that fragments of ancient viral DNA, once dismissed as mere junk, might actually play a crucial role in regulating our genes. This revelation comes from a novel method they developed to trace the evolutionary history of these viral DNA sequences, which had been overlooked in previous genome annotations. Guillaume Bourque, a lead author of the study and a professor in McGill's Department of Human Genetics, explains, by pinpointing which parts of our genome are unique to humans or primates and which originated from viruses, we edge closer to understanding what truly makes us human and how our DNA impacts our health and susceptibility to diseases. Remarkably, about 8% of our human genome is derived from viruses that infected our ancestors millions of years ago. These sequences, once thought to be useless, are now known to help switch genes on and off. The new study identifies specific sequences with regulatory potential, adding to the growing body of evidence that these overlooked sequences deserve more attention. But here's where it gets controversial. When the human genome was first sequenced 25 years ago, researchers identified viral DNA but lacked the tools for detailed study. With modern technology, the team revisited the genome and found many annotations to be outdated or incorrect. The researchers devised a new method that groups viral sequences based on their evolutionary history, rather than just sequence similarity. By tracing their evolution, they identified patterns suggesting which sequences might control gene activation or deactivation. Focusing on a viral DNA family known as MR11, the team discovered not 3, as previously thought, but 4 subtypes. One group, MR11_G4, was particularly active in human stem cells and featured a unique DNA motif found only in humans and chimpanzees. This group, the researchers believe, could be key in gene regulation. The current annotation of viral DNA in our genome shouldn't be considered final. It's high time we revisited and refined it, asserts Bourque. A clearer understanding of the genome could help scientists decipher genetic mutations linked to cancer and rare diseases, he added. About the study. The research, titled A Phylogenetic Approach Uncovers Cryptic Endogenous Retrovirus Subfamilies in the Primate Lineage, was authored by Sun Chen, Zhizong Zhang, Yizhi Yan, Clement Gubert, Guillaume Bourque, and Fumitaka Inoue. It was published on July 18 in Science Advances. The study received support from the Canadian Institutes of Health Research, the Canada Research Chairs Program, the Fonds de Recherche du Québec, Santé, and Japan's World Premier International Research Center Initiative. So, what do you think? Could ancient viruses in our DNA be more significant than we ever imagined? Share your thoughts in the comments. Junk DNA's vital role in gene regulation revealed. For years, the genome's so-called junk DNA has been a hot topic of debate, and for good reason. Labeled junk because it doesn't code for proteins, these vast stretches of non-coding DNA make up the majority of our genetic material. But what if these sequences aren't meaningless after all? What if they actually play crucial roles we've only just begun to uncover? This is where things get fascinating, and controversial. A groundbreaking international study led by biologists in Asia has shed new light on why certain non-coding segments might be essential rather than useless. Specifically, they've zoomed in on one particular region, revealing that it could be a key regulator in controlling cellular processes. This challenges the old notion that non-coding DNA is just genetic clutter. One major component of this non-coding DNA consists of transposable elements, TEs. These sequences are remnants of viral DNA that invaded genomes ages ago, embedding themselves into the DNA of entire species. At first glance, their viral origins might suggest they're unwanted passengers with no real purpose, akin to genetic freeloaders. However, evolution is a master recycler. TEs have been part of the genomic landscape far longer than humans have existed, and just because they may have started without function doesn't mean they've remained irrelevant over time. The name, transposable, comes from their remarkable copy and paste ability. These elements can duplicate themselves and insert copies into new places within the genome. This phenomenon explains why TEs take up such a significant portion of our DNA. But here's the twist: the machinery that enables TEs to replicate can also influence how other genes behave, effectively reprogramming cellular activities by repurposing these viral leftovers. Delving deeper, the researchers examined a gene known as MR11, a member of this TE family, by categorizing its various versions based on how they've been conserved through primate evolution. They identified 4 distinct groups or generations, ranging from ancient to more recent variants. Their findings were revealing. The newer MR11 variants showed much stronger ability to regulate gene activity, implying that natural selection has been fine-tuning this gene's regulatory function ever since it entered the human genome, likely through a viral infection. Intriguingly, the team went further to suggest that the expansion of such regulatory elements might be intimately tied to how new species evolve. Because regulatory elements can impact numerous genes simultaneously, even a small modification in these regions could spark significant genetic shifts, helping a population become genetically distinct. This raises an exciting question: could the activation and refinement of junk DNA be a hidden engine driving evolutionary leaps? Of course, not all scientists agree about junk DNA's value. While many now reject the idea that most of the genome is useless, some argue that much of it might still be selfish DNA, functioning primarily to propagate itself without benefiting the host organism. This study provides compelling evidence against that view, illustrating how evolution tends to co-opt and repurpose genetic elements that might have once been mere stowaways. It's important to emphasize that this research doesn't claim every piece of junk DNA is functional. Instead, it highlights the incredible diversity of roles that these non-coding regions could potentially play. Looking at stretches of DNA previously dismissed as irrelevant, the more reasonable assumption becomes that they probably hold functions we haven't yet deciphered. This discovery opens the door to rethinking countless genetic mysteries and challenges us to reconsider what makes up the fabric of life. So, what do you think? Could these viral remnants be the heroes we've overlooked in evolution's story, or are we reading too much into genetic noise? Feel free to share your thoughts below, because this debate is far from over. Unraveling the junk DNA myth, how it impeded science. The myth of junk DNA, a barrier to scientific progress. For many years, evolutionary biologists dismissed non-coding regions of DNA as mere evolutionary debris, a belief that significantly hindered researchers from exploring these poorly understood segments of the genome. However, since 2008, a wave of discoveries has dramatically shifted this perspective on what is often labeled as junk DNA. Numerous functions have been identified within these non-coding regions, with new findings emerging each year. In a recent episode of ID the Future, Dr. Casey Luskin highlights the achievements of 2 American biologists who were awarded the Nobel Prize for uncovering functions in what was once thought to be useless DNA. MicroRNAs serve as another compelling example of how the assumption that our genome is cluttered with worthless material has obstructed scientific advancement. When microRNA was first discovered in 1993, along with its crucial role in post-transcriptional gene regulation, it was met with skepticism and indifference from a scientific community that largely believed non-coding DNA was without purpose. The awarding of the 2024 Nobel Prize brings to light a poignant question: did the prevailing notion of junk DNA delay the recognition of a discovery worthy of a Nobel Prize? The evidence suggests that it did. Dr. Luskin also points out that proponents of intelligent design have long anticipated the functional significance of junk DNA, dating back to the 1990s. Notable figures like Richard Sternberg and William Dembski were among the early voices challenging the idea of junk DNA, echoing the sentiments of Jonathan Wells, who critiqued this concept in his 2011 book, The Myth of Junk DNA. In this ongoing debate, the intelligent design perspective has been repeatedly validated, most recently by the Nobel Committee itself. For those interested in delving deeper into this topic, you can listen to the podcast episode, here, https://idthefuture.com/2082. Explore further. Asterisk check out Casey Luskin's article, here, https://evolution-news.org/2024/12/2024-nobel-prize-awarded-for-the-type-of-function-for-junk-DNA that sparked this discussion. Asterisk discover more insights from Casey regarding the myth of junk DNA, here, https://idthefuture.com/1908